Hello and welcome to the last episode of the Revisited Season 2. This video will revisit apples. You'll first start with the history of the domestication of apples using a paper by Dr. Amadine Cornille, who used ancient DNA, preserved remnants of different apple species, and archaeogenomics to determine the history, domestication, and integration of apple. Let's get started. Using the genetic fingerprinting technology, specifically macrocylite markers, it can be concluded that apples were domesticated in Central Asia from wild Malus sieversii specimens based on genetic evidence, and from there gradually moved westward. The reason why we know this is because remnants of DNA found in more western species of crabapples such as Malus sylvestris have been found within both modern and preserved ancient apple DNA. From an archaeobotanical perspective, there's been several dig sites that showcase early use of apples by people, starting with wild apples, followed by increasingly managed wild stands, including cultivation and protection of these wild stands, and following that, intentional cultivation of apple trees. Although at this point the process of how this happened is patchy due to the fact that plant remains of apples such as seeds, fruit, wood, or pollen are quite rare within the dig sites. This is because much of the dig sites are dry, and apple remnants can only be preserved in a charred form in dry conditions. Whilst in waterlogged locations, plant remnants can be preserved a lot more easier, specifically for apples. Indeed, whole preserved apples have been found in waterlogged archaeological deposits, and these whole apples have been perfectly preserved within said wetland dig sites. What early dig site evidence obtained was a higher ratio of apple seed remains in Europe versus Asia, specifically wild apples, and gradually going into cultivated apples. This allowed for a reconstruction of how apples were disseminated in Europe. However, there are only a small number of finds in terms of apple cultivation in Kazakhstan, Mongolia, Nepal, Tajikistan, and Turkmenistan, and they only date to the past 3,000 years. This prevents us from tracing the history of earlier cultivation or domestication in this area. Unfortunately, based on all the evidence, this area is the origin point of apple cultivation. So using only archaeological evidence, we are unable to determine the full history of domestication within apples. Now, using ancient DNA obtained from apple remains in European sites, one is able to glean a little bit more information. 2 Neolithic and 2 Roman sites in the Alpine foreland within various European dig sites have obtained various degrees of different types of DNA, specifically chloroplast DNA and partially degraded remnants of nuclear DNA. They were unable to glean anything from the ancient chloroplast DNA, but nuclear DNA, however, showcased some interesting finds from the Roman samples of apple remnants. The DNA sequences that were able to be salvaged showcased both remnants of Malus sylvestris and Malus sieversii DNA, indicating that these apples are hybrids between the two. This apple remnant was found in the waterlogged remains, and the DNA itself was extracted from a seed from a preserved apple. Other dig sites ranging from England to Estonia also showcased useful data to determine the genetics of different apples. For instance, 2 Estonian samples of apple remnant with DNA intact showcased genetic similarities between both cultivated apples and a Russian specimen of Malus sylvestris, indicating that Estonia's apples are derived from Russian apples. So it's likely that apples came to Russia before they came to Estonia in ancient times. There have also been heavy advances in archaeogenomics. Thanks to the presence of waterlogged and desiccated plant materials preserved within wetland dig sites, a greater level of the genome within the plant material can be obtained much easier. And there have been great advances in the production and development of plant archaeobotanical DNA extraction protocols. As of right now, new technologies are being introduced to archaeology that are also used in common DNA sequencing program. The latest one is called the target/capture enrichment method. This new method reduces contamination risks and targets several genomic spots on a piece of DNA at once. Through this process, it is hoped that the genes for water and nutrient use efficiency, dormancy, flowering time, fruit ripening, color, acidity, and disease resistance will be uncovered from this process. Now on to the full conclusions that the researcher comes to based on the information she has gathered. The use of these genetics, specifically population genetics and archaeogenetics, have showcased a great deal how the apples were domesticated and spread by people over the course of generations in history. Genetic analyses support a Central Asian origin for the cultivated apple, specifically Malus sieversii, with secondary contributions from other species such as the European crabapple Malus sylvestris. The genetic studies also showcase weak spatial population structure, which means that the wild apples have genetics from cultivated apples and vice versa, with high levels of gene flow between both groups. This indicates that a major portion of the domestication process was continuous introductions of new DNA from other wild species into the primordial form of the domesticated apple. Genetic analysis of the cultivar Golden Delicious showcases transposable elements, which are a type of gene that can bounce from one part of the genome to another at a relatively frequent rate, adding new genetic diversity. This transposable element first appeared, based on the genetic analysis, during the uplift of the Tian Shan Mountains, indicating that this trait is a very ancient trait. One of those transposable elements is actually a gene called MDMYB1, which actually is used in making the red coloration, found on the skin of different apples. This indicates that the red coloration of that particular apple evolved during the time of the uplift of that mountain range. What this means is that the trait for red coloration is derived from the Tian Shan Mountain Malus sieversii population, not from outside sources such as Malus sylvestris. Another interesting factor is that the large fruit genes were actually found fixed within the wild populations of Malus sieversii as well as domesticated forms, indicating that natural selection fixed that trait before domestication even occurred. This makes sense considering that in the previous video I made on apples, indeed my first video I've made, it was mentioned that a switch of seed dispersal methods from birds to bears led to natural selection selecting for larger fruit to appeal to bears, as well as sweeter fruit, which also appealed to bears. Now back to hybridization. As was mentioned before, the incorporation of other wild species into domesticated populations was a major player within the domestication of this crop. Most of this seems to have happened across Silk Road when plant materials were transferred from Kazakhstan to Persia and from there to the rest of the world. In modern days, Malus baccata, or the Siberian crabapple, has been used extensively to breed new varieties of apple. It has been specifically used in China extensively due to excellent resistance to cold stress and apple scab. People have likewise been experimenting with Malus floribunda, Malus hupenstris, Malus zumi, and Malus X robusta to breed new varieties, to develop apples that are adapted to specific stresses and climates. For instance, they found resistance traits within Malus robusta to fire blight. Historically speaking, Malus sylvestris and Malus domestica lack the genetic and reproductive barriers to prevent continuous crossbreeding. And to this date, indeed a paper by Dr. Gargiania showcased various levels of affinity between different species, indicating that hybridization occurred all across different trade routes. Many of the cultivars found in the Balkans have minor traces of Malus baccata in their lineage, alongside several of its subspecies and hybrids. In the east, remnants of across the Middle East and all parts of Asia, most of the apples have traces of Malus orientalis pedigree in their lineage. Many of these cultivars could be traced back to the Roman Empire and Persian Empire in the 7th century BC. So a wide array of different hybridization events occurred through possibly purposeful human intervention and random pollinations. Various bits and bytes of genetic material from other apples have been infused into the gene pool of the cultivated apple. This caused Dr. Gargani to propose that Iran is one of the centers of diversity for apple domestication. Now we move on to more integration when we talk about red-fleshed apples. Red-fleshed apples are apples with pink or red flesh, rather than the typical white or yellow flesh found in most apples. A paper by Dr. Nan Wang wanted to figure out the origin, flavonoid synthesis mechanism, and breeding of red-skinned and red-fleshed apples, and here are the results. Based on current genetic records, the subspecies of Malus sieversii subspecies Nequitiania, is the origin point of red-fleshed apples. Indeed, in most of the Malus sieversii specimens, it's been found that there's greater levels of flavonoid content in the wild Malus sieversii than in modern apples such as the Starkane apple variety. Now the full genetics and biosynthetic pathway is way beyond the scope of my understanding since biochemistry is not my forte. I will however link the paper in the description below so you can look it over. But based on current information, it's likely that mutated genes MD-MYB10 and MD-MYB11A genes are responsible for creating the red-fleshed trait. The breeding of red-fleshed apples, however, can be traced back to 1897 when a Mr. Hansen encountered an unusual apple in the forests of Turkmenistan. This became known as the subspecies Nequitiania. During this time, he bred several different varieties of red-fleshed apple like Almata. The breeding program continued when an old apple variety was rediscovered called Surprise. It's an apple with pink flesh. Albert Eder, up until 1944, bred a great deal of apples using this variety as a parent. Although Pink Pearl is the only commercially released variety, the Almata and Surprise lineage of apples lay the groundwork for all the red-fleshed apples we see today. One thing that is common between both lineages is that both the skin, the flowers, the bark to a certain degree, and the leaves in fall are red, as is the skin of the fruit. In England, the Eder apples were obtained and then marketed under the Rosette series, such as Pink Pearmain, Blush Rosette, Thornberry, Rubaiyat, Christmas Pink, Grenadine, and Pink Parfait. In Japan, various hybrids between Pink Pearl and other apple cultivars occurred in Japan, whilst the Almata types and recreations of the original cross have been used extensively in Germany. Other researchers right now are studying the possibility of using wild Malus sieversii in the breeding of new forms of red-fleshed apple to add disease resistance into red-fleshed apples. We'll conclude this video with a talk about the red-fleshed lineages in particular. I've had several of the red-fleshed apples, and I've made reference to several other videos regarding red-fleshed apples, one of which will be linked below in the description. I've had apples from the Surprise lineage and the Almata lineage. First things first, the red flesh only extends to the point slightly above the skin of the apple, i.e. the red flesh doesn't extend beyond the first inch of skin. Number 2, having had some of these Almata lineages, the flavor is not that good. The flavor is more akin to a really bad Macintosh. Although that's just my opinion, you might find it tastes a little bit better. The flavor of the Surprise lineage is much more to my liking. I've had 2 apples of that lineage, Pink Princess, not to be confused with Pink Lady, and Arlie's Red Flesh. The former tastes like fruit punch and the latter tastes like strawberry lemonade. But in both cases, the red flesh lineages tend to be very susceptible to disease, especially the Surprise lineage. Although there have been improvements such as Pink Princess, which is scab resistant. The one overarching theme within both red flesh apple lineages is mealiness. The apples are incredibly mealy, although in the case of the Surprise lineage, I would say the flavor overcomes that quite extensively. But to further improve the varieties of red flesh apple, the Almata lineage would have to incorporate a lot more better flavored apples to improve the overall sensation and flavor of these lineages. And the mealiness and lack of disease resistance, especially to scab and fire blight, will have to be overcome in order to make red flesh apples a viable crop going forward for organic growers, and for commercial orchardists in general. That about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.